AS Level Biology 9700 Paper 2 Variant 2 October November 2025, and in this video we are going to discuss question number 1. And question number 1 is: the wall of the small intestine is highly folded to form villi. Between the villi are infoldings known as crypts of Lieberkühn. So figure 1.1 is a diagram of a section through a single villus and a crypt of Lieberkühn, and you can see the different labelings over there, that is lacteal, capillary network, enterocyte, goblet cells, enteroendocrine cell, and then Paneth cell. Crypt of Lieberkühn, you can see this folding, an intestinal stem cell, venule and lymph vessels, and one labeling axis there. Now we move towards the question. Figure 1.1 shows that the epithelium of the villus contains many goblet cells and cells known as enterocytes. Both cell types have microvilli on the apical surface. Apical surface means the surface facing the gut lumen. Goblet cells are involved in the production of mucus. So enterocytes are adapted for the absorption of the soluble products of digestion. So these products enter the circulatory system. So a student incorrectly stated that an enterocyte has many cilia on its apical surface. Explain the difference between a cilium and a microvilli. So we have to discuss the difference. Marks are two, so we have to one or two differences between them. So first of all, I am going with the structure. So the difference between these two structures are: cilia are hair-like projections, and they contain a complex arrangement of microtubules, 9 plus 2 arrangement, and are capable of movement. On the other hand, the microvilli are finger-like folds of the cell surface membrane. They contain active filaments and are non-motile. They cannot move. So the function of both of these, if we want to write the difference in the function, so the primary role of microvilli is to increase the surface area for the absorption of nutrients, and cilia are typically used to move substance like mucus or an egg cell across the surface of the epithelium. So we have to write these two differences in structure and function both. But in exam, you can choose any one of these because it is of 2 marks. So first of all, the differences which I have told you about the structure. So the cilia are hair-like projections that contain a complex arrangement of microtubules. As you had read in class, 9 plus 2 arrangement. So, and are capable of movement. Microvilli are finger-like folds of cell surface membrane. which contain actin filaments and non-motile. And the function microvilli increase the surface area. For absorption. Cilia are used to move substances like mucus across the surface of epithelium. So moving towards the part B of this question, it was very simple. You must know all these. Suggest one role of the mucus produced by the goblet cells of the villus. There are many, you can say, functions, or you can say the role of this mucus. So we have to write one. So we can write any one. It lubricates the lining of the intestine to allow the smooth passage of food. And second one, we can write that it acts as a protective barrier for the intestinal wall against digestive enzymes or acidic contents. It also helps to trap the pathogens to prevent infection. We can write any one of these. So I would like to prefer the it acts as a protective barrier. Protective barrier for the intestinal wall against digestive enzymes. Or acidic content. Second one, it helps trap pathogens or microbes. Part C, in figure 1.1, blood vessel X delivers blood to the capillary network of the villus where tissue fluid is formed. Some of the fluid passes back into the capillaries and then into the venule. Blood vessel X receives blood from an artery. Name the type of the blood vessel represented by X. So we are moving towards the X. What is X? Yes, X is there in this figure. X. This is X. So you can see the X is a blood vessel receives blood from an artery. So it is receiving the blood from an artery, So that is uh, and delivers it directly to the capillary network. So this is the capillary network. So it's delivering over there. So it is an arterioles. We will write its name on the given space. So it is arteriole. Now the next part, second part of the question, part C, some part of the describe the formation of tissue fluid in the capillary network of the villus. Tissue fluid. Tissue fluid, is formed through a process of pressure filtration. So we have to describe its formation. So how do it is formed? So students, at the arterial end, arteries have high pressure. So at the arterial end of the capillary near vessel X, the blood pressure, which is also called as the hydrostatic pressure, is higher than the osmotic pressure. Osmotic, so this high pressure forces water and small soluble molecules out through the tiny gaps in the capillary walls. You know this. That is the tiny gaps are present there. So large structures like red blood cells and plasma proteins are too big to pass through the gaps and remain inside the capillary. So I would like to write this answer on the given space as it is of 2 marks. At the arterial end of the capillary, the blood the blood pressure or hydrostatic pressure is higher than the osmotic pressure. So this high pressure forces water and small soluble molecules and out through the tiny gaps in the capillary, which enters into the tissues and it is called as tissue fluid. So large such structures like red blood cells and plasma protein proteins are too big to pass through. Pass through the gaps and remain inside. Because they are larger in size, so they are unable to pass. So part C is completed. Now part D. And part D is in response to the presence of compounds in the gut lumen, enteroendocrine cells synthesize and release peptides that are single cell signaling molecules. One of these cell signaling molecules is known as GLP-1. So GLP-1 initiates a number of responses in different body cells. Some of these responses include the increase in release of the hormone insulin from cells in the pancreas, the decrease in the release of acid from cells in the stomach. So outline in sequence the main stages involved in the process of cell signaling by GLP-1. So sequential outline can be this, that is, first of all, GLP is released from enteroendocrine cells into the blood. It travels as a ligand through the circulatory system to reach the target cells, such as those in the pancreas or the stomach. Then it binds to a specific receptor located on the external surface of the cell surface membrane of the target site. So it is a peptide bond. So it is water soluble. It cannot cross the phospholipid bilayer. So it must bind to a transmembrane receptor. So the binding causes a conformational change in the receptor. So this change activates an intercellular signaling. The signal reaches a target protein and uh, protein or organelle within the cell, triggering a specific physiological change. In this specific case, the response is either the secretion of insulin from the pancreatic beta cells or the, you can say, inhibition of the acid release in stomach cells. So we will write the sequence because it is of 3 marks. So it need little bit more words to write. So GLP-1 is released from enteroendocrine cells. It travels towards target cells. It binds to a specific receptor located on the external surface of cell. So it needs a receptor. This is compulsory. The binding pause or conformational change in the receptor which activates intracellular signaling. The signal the amplification of the signal takes place and it reaches the target protein. which which triggers the specific response, which can be the increase in the release of the hormone, or which can be the decrease in the release of acid from cells in the stomach. Any one of these, according to uh, these changes. Now, the next part E is there to discuss. Pinna cells, which are secretory cells, are located between intestinal stem cells at the base of the crypt of Lieberkühn. As shown in figure 1.1, a penny itself has a very different appearance to an intestinal stem cell. In 1.2 is a transmission electron micrograph of a penny cell. You can see it is magnified. Now, moving towards the next question, part 1 of this. Pinnate cells are formed following the mitotic division of an intestinal stem cells during a cell cycle. So complete the cell cycle. You can see the interphase, cytokinesis, and mitosis. After interphase, mitosis leads to cytokinesis, in which the division of cytoplasm takes place and interphase occurs. So we have to write the name of stages of the mitosis. So the different stages of mitosis, they are prophase, prophase. Let me write it again. Prophase, metaphase, anaphase, and the fourth one is telophase. Now the part 2. One of the functions of the pineal cells is to synthesize and secrete peptides and proteins that act against pathogens in the gut lumen. State and explain the evidence visible in figure 1.2, which suggests that a pineal cell is a secretory cell. Synthesis may synthesize many peptides and proteins. So first of all, we will go with this question, that is the we have to provide the evidence, state and explain the evidence. But first of all, we have to state and then we have to explain. So first of all, we will explain that the presence of many secretory vesicles are likely located near the apical surface, the top of the cells. And if we want to explain it, is that is these vesicles store and antimicrobial peptides and before they are released into the gut lumen via exocytosis. So if we talk about them, these you talk about these prominent which Golgi apparatus, which modifies and packing these proteins into the vesicles. So the evidence for synthesizing many peptides and proteins. So an extensive network of rough endoplasmic reticulum is there. So the rough endoplasmic reticulum is covered in ribosomes, which are the sites of protein synthesis. A large amount of rough endoplasmic reticulum indicates the cell is dedicated to producing high volume of proteins for export. So we can write all this, that how do it takes place. First of all, the statement, the presence of many secretory vesicles likely located near the apical surface. So this is the statement. So evidence, these vesicles store the anti antimicrobial peptides and proteins. Before they released. So this is one of them, and the other statement can be the rough and extensive network of rough endoplasmic reticulum and the explanation for this evidence is the rough endoplasmic reticulum is covered in ribosomes. Ribosomes, which are the sites of protein synthesis. endoplasmic reticulum indicates the cell is producing high volume of protein. Now, the next part or the last part of this question, explain why a pineal cell has a very different appearance to an intestinal stem cell. So, reason behind this is that a pineal cell is a fully differentiated cell with a specific function, which is secretion requiring specialized organelles like are rough endoplasmic reticules and granules. In contrast, an intestinal stem cell is undifferentiated and its primary role is to divide to replace other cells. So it lacks the extensive secretory machinery seen in the beneath. So this is the only one difference which we can write. Fully differentiated cell with specific function. In contrast, an intestinal stem cell is undifferentiated is undifferentiated and its goal is to divide. So it was all about question number 1. Please like, share and subscribe my channel. Thanks for watching.